Initiate replication, the initiator protein locates and binds to the origin of replication and starts to unwind DNA at the origin. Helicase then attaches to the replication fork and moves along the fork, unwinding it like a zipper or a can opener. The orange blob represents gyrase, which moves ahead of the helicase on the still coiled portion of the DNA to prevent torsional strain and supercoiling. The gold stars represent single-strand binding protein that then attach to the unpaired DNA strand to prevent it from recoiling. Elongation is the second stage of replication. RNA primase starts this by laying down a primer which provides the first 3' OH that DNA polymerase III needs to link to start synthesizing DNA by adding nucleotides. DNA can only be added in the 5' to 3' direction, so one of the strands is able to race down the track and replicate no problem. This strand is called the lead strand. The other strand, or the lag strand, must have primer laid down as more of the track is available to attach to still move in the 5' to 3' direction. Because of this, DNA polymerase III must stop when it catches up with the previous primer. These segments of DNA are called Okazaki fragments, and to me they resemble the cars on a train, with the RNA primer being the connecting links. As the DNA continues to elongate, DNA polymerase I goes through and removes primer and fills in the gap behind it by moving in the 3 to 5 direction, but cannot completely connect these fragments and leaves nicks between. These nicks in the track of sugar-phosphate bonds that were not able to be filled by DNA polymerase I are then sealed by DNA ligase by adding a phosphodiester bond in between. This process will be repeated until the entire genome is replicated, which is not very extensive for a prokaryote. If any DNA polymerases stopped working right, the organism would die. But specifically, if DNA polymerase I stopped its 5 to 3 exonuclease activity, then the RNA primer would remain in the DNA genome, which would also kill the organism.